Welcome, ladies and gentlemen, to the SelectBio Extracellular Vesicles webinar series. We're very pleased to welcome Spectradyne as the sponsor of today's webinar. It's going to be an exciting webinar with a lot of great speakers. Before I get started with introducing the speakers, let me just mention that Spectradyne is a US company which was founded in 2013 that has commercialized a powerful new technology for counting and sizing single biological nanoparticles such as extracellular vesicles and viruses. So it's very timely, this webinar. You'll learn more about it, about the technology and how it can be used today. We have three excellent speakers. The first speaker will be Jean-Luc Fréquin, who is the CEO of Spectradyne. After Jean-Luc, Mike Paulaitis from Johns Hopkins will speak. And then finally, Josh Welsh from the NCI will present. And also, we're happy to take your questions. So if you would kindly type in your questions in the question box, at the conclusion of the presentation section of the webinar, we'll kind of go through each of the questions and the speakers will address them. So without any further ado, it's my pleasure to welcome Jean-Luc to the podium and for him to deliver the first presentation. So Jean-Luc, it's all yours.
Great, thank you, Nam. Hello, everyone, and thank you for attending our webinar today. I hope you'll enjoy listening to our guest speakers who will talk about the importance of careful EV concentration measurements in their own research. I will kick us off with an overview of EV quantification methods, including Spectradyne's own NCS1, and then turn the mic over to our two invited speakers, Dr. Paulaitis from Johns Hopkins University School of Medicine, and then Dr. Welsh from the National Cancer Institute. And before we get started, I'd like to say thank you to both of our speakers for sharing their precious time with us today so we can learn about what they're up to. So first of all, why are we all here? I think we all understand that EV concentration is a critical experimental variable, and the tighter we can control our experimental variables, the tighter our experimental outcomes will be, and the better science ultimately we can do. For EVs specifically, this comes up in two main application areas. One is when EVs are being considered or used as therapeutics. This is essentially a drug delivery application. And in this case, the concentration of EVs is really just the dose of the therapeutic itself. And so obviously we have to control the dose well, because that leads to safer, more effective products. It also helps when we're evaluating different formulations or different preparations of these therapeutics to perform tighter experiments this way. The second application of EVs where concentration is important is when we're using them as an assay readout. And in this case, as an example, is a biomarker detection assay. So in this case, the concentration is the test result. And so the better we can control these experimental variables and the concentration measurement, then this will lead to better sensitivity, better specificity, and ultimately better assay. So essentially, the more accurate concentration EV measurements we can make, then the tighter experimental outcomes and the better science we can do. So how are people usually measuring their EVs right now? So I think most of us would probably agree that cryo-EM is the most powerful EV characterization technology. It's a gold standard. You get to see what you see what you're looking at, and distinguish EVs using the lipid bilayer, et cetera. So, however, obviously it's impractical for routine use as much as we'd all like to have one in our labs, right? NTA, nanoparticle tracking analysis, has been around for a long time. This is an optical technique and has been instrumental in developing the field of EV science. Although lately, I think researchers are increasingly understanding its limits. And one in particular that Spectradyne likes to talk about a lot is that its limit of detection depends really on the sample that you're trying to measure. And so we need better techniques. Flow cytometry is a really powerful technique, especially when it comes to phenotyping different particles and large particles. it is only recently started pushing into smaller particles, and I think even Dr. Welsh, a flow cytometry expert, would agree with me that in a heterogeneous sample, it's impossible to get accurate size concentration of the particles because it depends on, you have to assume the optical properties of the particles, and if you have a mixture, you can't do that. We can talk about that more later. In terms of non-optical techniques, there has been tunable resistive pulse sensing, which I expect most of you have heard about before and maybe used. This has been around for a long time also, as EV field has been developing. It's a good approach. Spectradyne likes the approach a lot, but from what we hear, it's a challenging implementation. This is where Spectradyne comes in. We've developed microfluidic resistive pulse sensing, or MRPS, that is rapidly emerging as a powerful technology because it's fast, affordable, and easy to use. And so because Dr. Paulaitis and Welsh will be showing data from the NCS1 or MRPS system, I will spend a bit of time talking about how it works next. But before I do, I'd like to draw your attention to a recent preprint that is now up on the archive. This is from researchers at Johns Hopkins University and NIST, including Dr. Paulaitis, who will speak in a minute. This is a very solid, even-handed assessment of 4 new single particle EV analysis platforms, and I encourage you to go and check this out. It's open access, obviously. So Spectradyne's NCS1 is a benchtop instrument. It's about 1 foot wide, and it delivers accurate concentration and size of biological nanoparticles starting about 50 nanometers diameter up to about 10 microns diameter. Importantly, it's an electrical technique. It's non-optical, so it doesn't suffer limitations of optical techniques. And it leverages the power of microfluidics to deliver a bunch of important features, including making it fast and easy to use, and only requiring 3 microliters of your sample to measure. Because it's a non-optical technique, it lends itself to diverse applications. Obviously, today we're talking about extracellular vesicles, but many of our users use the technology to quantify virus and phage. And in that application, the NCS1 delivers virus concentration in minutes without having to wait for biological titer that can take hours or days. And this will be a topic of an upcoming webinar, so please stay tuned for that. Other scientists that use the NCS1 use it for quantifying protein aggregates, non-viral gene therapy vectors, lipid nanoparticles, etc. The list is long, and you can learn more about some of these applications on our website if you visit our library, where we have links to peer-reviewed publications and interesting measurement examples. So the NCS1 works as follows. Using microfluidic resistive pulse sensing, particles are counted, analyzed one by one, and it measures really directly the concentration and size of the particles. So what we do is we flow the sample through a bottleneck in the fluid flow, in the fluid channel. And every time a particle goes through the bottleneck, the constriction, we measured essentially 2 things about it. One is the particle size. So this is a plot showing the electrical signal we get from a single particle measurement. And we measure 2 things: the particle size, that's related to how big this peak is, and how quickly the particle went through the constriction. And that's important because it tells us the flow rate of sample through the constriction. So this is the measurement of a single particle. If we measure longer, this is what the data looks like. And here we've highlighted all the particle events with red dots. You can see there are particles being measured at different sizes. And straight away from the histogram of the counts, we can see that there are 4 different populations of sizes in the sample. And now, because we know the volume of liquid that we've measured from measuring the transit time of the particles through the constriction, we take the number of counts divided by the volume, and that's the concentration very directly. So this is an example of the final output from our system. It's a graph showing particle concentration on the y-axis versus particle diameter on the x-axis. This particular sample that I showed you, and the analysis is here, was a mixture of polystyrene particles, 50, 90, 120, and 150 nanometers in diameter. You can see that all the different populations were well resolved, and we can be quantitative over any size range in between here. So this combination of accurate concentration and size and the ease of use of the system is not currently available in any other technology on the market. I'll just show you two examples of how well it works. This is a measurement of a retroviral drug formulation, serial dilution, and you can see in the data, this is again concentration versus particle diameter. There's a peak in the distribution where the viral particles are over a broad background of other particles. And as you can see, when we dilute the sample, the concentration goes down, and we get a nice linear response for this measurement. One more example I'll show you is a measurement of a liposomal drug formulation. This is the measurement of a single sample in 3 different size ranges, and you can see the measurements on the different size ranges agree very well in the overlap regions. And in particular, what I want to draw your attention to is that this sample, the particles in this sample span over 9 orders of magnitude in concentration across between 65 nanometers up to 5 or 6 microns in diameter. So there's a very broad dynamic range of this technology. So Spectradyne's benchtop instrument helps you do faster and better science, and we want to help you too. We want to know how we can help your science. So one of the ways that we demonstrate the power of this technology is to measure samples for free for researchers. So please get in touch with us and send us one of your samples so we can help you or work with you. At this point, I will turn over the microphone to Dr. Paulaitis from Johns Hopkins University, who will speak about the importance of EV concentration measurements in studies of macular degeneration. And then we'll hear from Dr. Welsh from NCI after that. Thank you very much. Enjoy the speakers.
Thank you, Jean-Luc. I just want to make sure everybody can hear me and that you can see my screen.
You can go ahead and show your screen now, Mike.
Okay.
All right.
Can you see it?
Yes.
Okay. So good morning, everyone, and thank you, Spectradyne, for inviting me to give this talk and share my experience as a user of this instrument, and hopefully to make contact with other people out there who are also using the instrument to share their experiences. So the title of my talk today is the importance of EV concentration measurements on in our studies of age-related macular degeneration. My collaborators on this project are Ken Whitwer and Emily Malik in the Molecular and Comparative Pathobiology Department at Johns Hopkins. Emily is in Ken's group, and she performed all the SP-IRIS assays that you'll see in this talk. And Dr. Jim Handa is my collaborator in the Department of Ophthalmology. Age-related macular degeneration is a leading cause of vision loss in people over 50. The macula, the most sensitive part of the retina, is damaged in AMD, and the results, as you can see in this diagram to the right, is blurred vision near the center of the visual field that progressively grows as AMD progresses. The scope of the problem, about 196 million people are afflicted globally with AMD. It's projected to be 288 million in 2040 with our aging population. And in the US, the number of people with AMD is comparable to the number of people with all the invasive cancers, as well as more than double the number with Alzheimer's disease. The principal cell type implicated in AMD is the retinal pigment epithelial cells, or RPE cells. These cells form an epithelial monolayer in close contact with photoreceptors. And you can think of this, this monolayer as the blood-retinal barrier, analogous to the blood-brain barrier. The RPE cells phagocytose approximately 30,000 photoreceptor tips daily. This represents a significant physiological load on both the lysosomal and exocytotic pathways of RPE cells. It's a highly energy-intensive process and requires highly functional mitochondria. With aging, mitochondrial function declines, the physiological load of the RPE cells increases. There's progressive deterioration of RPE function, and this leads to photoreceptor death and vision loss. So the research hypothesis we have in studying AMD is that RPE cells under oxidative stress release extracellular vesicles that have small RNA profiles that reflect this diminished mitochondrial capacity. These profiles can predict progression from mitochondrial dysfunction to disease onset and identify potential targets for inhibiting the spread of AMD. To date, we've looked at two cell lines in this study. One is the ARPE-19 and immortalized RPE cell line from ATCC. And the objective of this first study was a proof of concept to show that mitochondrial microRNAs in EVs are released in greater numbers when the ARPE-19 cells are mitochondrial distressed. And we do that by treating the cells with Retinal, which inhibits the electron chain transfer in the mitochondria. The second set of cells we looked at were induced human pluripotent stem cell-derived RPEs, fully differentiated that form monolayers. And the objective of this study was to look at the apical versus basal release of EVs and their content containing both microRNAs and micro-tRNA fragments. Oops. So the two workhorses we use in these studies are the Spectradyne's NCS1 instrument to determine average vesicle size, the EV size distribution, and to obtain a reliable estimate of the number of concentrations of EVs released per cell when we impair the mitochondria in these cells. And then the other workhorse instrument we use is the ExoView platform from NanoView Sciences, the SP Iris, and the SP Iris with fluorescence. So this shows you some of the data for the ARPE-19 EVs. This is capturing the EVs on the ExoView chip, both for no treatment and Retinal treated cells. And we see that predominantly the highest efficiency is for CD81, but we see a population across all tetraspanins, which are characteristic of EVs. Fluorescence labeling these captured EVs, both no treatment and Retinal treated cells, shows that EVs release CD81, CD9 co-expressing EVs in almost equal amounts, and this is independent of the treatment conditions. We can confirm both CD81 and CD83 by Western blot, and we can also show the presence of syntenin-1 and internal cargo within these EVs, which allows us to make the observation that both EV populations, from the treated and untreated cells, contain vesicles of endosomal origin, or essentially exosomes. Size distributions, number concentrations are obtained from the Spectradyne instrument. This shows you a typical assay we do. The TS400 cartridge is our workhorse cartridge, and the solid line then shows you a dynamic scaling model fit of that data. There's several important questions we want to answer by measuring these size distributions. The first one is, how many populations of vesicles do we see? And in particular, the scaling behavior is important as we go out to much higher diameters to see if there are apoptotic bodies or other particles present in our samples that would affect our results. And then to obtain average vesicle sizes and answer the question of how many vesicles are released in response to mitochondrial dysfunction, we want to know how to predict the area under this curve, and that requires us to know something about this peak and the minimum vesicle diameter. I'll talk about this in more detail in the second part of the talk, But let's look at some of the results that we have for the ARPE-19 cells. Let's start with the top left-hand corner, which shows our TS400 measurements and the scaling behavior fit to that result, which we've extended now down to approximately 35 nanometers in diameter, and compare that to what we see in terms of the scaling behavior from the Spectradyne TS150 cartridge. These, I think, are still experimental cartridges. We were fortunate enough to get a few of these from Spectradyne early on in our measurements. And the point I want to make here is extrapolating this data. As long as we have reliable statistics for this TS400 cartridge, we can accurately extend this scaling behavior down to much lower diameters. To answer the question of are there larger bodies in these samples, we can do measurements with both the TS400 and the TS2000 cartridge, and you can see from the blue lines that we detect no larger particles relative to the spike ends, which are polystyrene beads in both cases. You can also compare our size distributions to other measurements. In this case, we looked at asymmetric flow, field flow fractionation, multi-angle light scattering, which is a model-dependent measurement of the size distribution that also separates the particles by size. And again, we obtain, although there's a shift in diameter, a single unimodal distribution. And finally, we can just look at the samples. Do we see apoptotic bodies? Do we see mostly the characteristic cup-shaped morphology of the vesicles by cryo-TEM? So within the measurements we've done for the ARPE-19 cells, we can make the following observations. We obtain a unimodal size distribution. The model fit tells us the average diameter is about 63 nanometers of this distribution. The SB iris identifies a population of exosomes within this size distribution of particles we measure. And within the extension of the model, scaling behavior down to lower diameters, we measure approximately a threefold increase in the number of vesicles released per cell when the cells are mitochondrially distressed. I haven't shown this data, but we've also measured approximately threefold increase in mitochondrial microRNA miR494-3P as we distress these cells with, or impair these cells with rotenone. And those last two observations allow us to make these two conclusions. One is that the ARPE-19 cells with impaired mitochondria release more of this mitochondrial microRNA because the cells release more vesicles. And that when we induce mitochondrial dysfunction in these cells, more vesicles are released. So essentially we've linked vesicle release to mitochondrial dysfunction within these cells. So the second part of this talk is about the apical basal release of these EVs from these induced RPE cell lines. This represents a greater challenge to measuring the number of EVs released per cell because this is a polarized monolayer that loses the polarization with mitochondrial dysfunction, and it's exactly the asymmetry of EV release that we're focusing on as the cells lose polarization. Again, here's the SP iris results for EVs released apically and basally from these monolayers. And what you can see independent of treatment, apical release is about 2 to 3-fold higher than basal release. Fluorescence tells us there's very little difference between these different populations of tetraspanins. I show the results here for the apical EVs versus basal EVs. results are similar under no treatment conditions. And we now have the ability to permeabilize the captured vesicles, and we can then detect directly on these chips for the presence of syntenin 1. We see again syntenin 1 present in all these samples. So from these measurements, we conclude that the vesicles we're seeing are essentially the same, apically, basally, and independent of treatment. So what does SP-IRIS tell us about the size distribution that we can get from the label-free measurements? And these results show that the size distributions for apical versus basal release in both control and rotenone conditions are not too different. There's a peak around 50 to 55 nanometers, and the distribution falls off rapidly as we get to a diameter of about 100 nanometers. We can also plot this as we would the Spectradyne results. Oops. And this shows us again that these distributions are quite similar across all four measurements. They're a little more scattered in the basal release and the control conditions that could be experimental, as well as sample preparation. So how does MRPS tell us these distributions differ, apical versus basal release? Well, first of all, we can compare MRPS to SP-IRIS if we assume a total concentration of vesicles, and I show results here for CD63 capture. And you can see that the essential shape of these two distributions are in very good agreement. So in fact, the SP-IRIS is telling us the distribution is similar to what we're getting from MRPS, but it doesn't tell us the actual amounts of vesicles that are being produced in each case. From the Spectradyne measurements, we can get those absolute concentrations, and this shows results for no treatment conditions using, again, the TS400 cartridge. Apical release shows a larger population of vesicles compared to basal release, where both of these measurements are standardized by the addition of polystyrene beads, and the inset shows this extrapolation down to lower vesicle sizes using the TS300 cartridge. This graph just shows us that there's really no effect of adding these polystyrene beads on the distributions that we measure from MRPS. Rotenone treatment, we see different behavior. We see that the apical and basal release of these vesicles are essentially not different from one another. We've essentially lost polarization of these cells, and we've lost this asymmetry in release. To get the particle number concentration, then we have to extend that TS400 cartridge measurement down to lower diameters using the TS300 cartridge measurements, as I showed in that figure inset. That extrapolation follows quite well a power law distribution. And in fact, then we can model fit these results with that power law distribution. These are fit to the data the overlapping data of the TS300 and the 400. It shows a maximum of around 30 to 35 nanometers in the distribution and a minimum diameter of around 20 nanometers. And you can see from this plot that this part of this, the area under the curve here, is very important, and accurate estimates of that area are essential to get good number concentrations. So one thing we can ask is, if we can get the scaling law behavior well, what is the minimum diameter we really have to go to get an accurate estimate of the area under the curve? And we can look at results we've obtained from cryo-TEM. These are on cancer cell lines, but what they show for cryo-TEM images, that we can actually measure vesicles that are on the order of 20 to 25 nanometers in diameter at about the same frequency that we order or we measure or observe vesicles on the order of 100 nanometers in diameter. So that 20 nanometer model fit of the data for the minimum vesicle diameter is a reasonable one. Well, the fact that all four of these vesicle distributions look the same, that they have roughly the same scaling behavior, allows us to make a reasonable estimate of the total concentration of the vesicles. And this is what we find for apical versus basal release, that there are about threefold higher numbers of apically released vesicles versus basal release vesicles under control conditions. And when the cells lose polarity due to rotenone treatment, the number of apical vesicles goes down appreciably, and the number of basal vesicles remains the same. Again, the error bars are large, and this is a reflection of how well we can estimate that scaling law behavior down to lower diameters. Once we have the number of vesicles released, we can start looking at cargo, and we can make some preliminary observations here. Basal EV release contains EVs that have a higher protein content. Apical release is favored. If we look at characteristic microRNA, miR-494 is a predominant microRNA in RPE cells. And this shows that the polarized release of 494, which favors apical release, is lost when the cells are treated with rotenone, both QRTP and RNA-seq assays. And we can also look at tRNA fragments, which show the opposite behavior, that with loss of polarization, there's actually higher relative levels of these mitochondrial tRNA fragments in the basal versus the apical released EVs. So I won't read the conclusions here in this preliminary study. The 2 points I really want to make, though, are given in the bulleted items at the bottom. And one is accurate EV concentration measurements or reasonable estimates of total EV concentrations require extending current measurement capabilities of EV size distributions to smaller diameters. In these studies, we've used the instrument from Spectradyne to obtain characteristic scaling behavior to estimate these extrapolated distributions. And I believe the instrument has the capability over other instruments to do this with reasonable accuracy today. So with that, I will end and turn this over to, I guess, Josh.
All right. Josh, you're now the presenter.
Okay, hello everyone. Thank you to Spectradyne for inviting me to talk today. So today I just kind of wanted to take you through our experience with using characterization techniques, orthogonal measurements, and why they've been very important to us, and why we kind of recommend that all people doing EV studies use orthogonal measures. So just to give you some background to our work, I'm currently in the translational nanobiology section led by Jennifer Jones at the National Cancer Institute. And what we're attempting to build is a high throughput scalable clinical pipeline. So with the state of our field at the moment, this is obviously fraught with challenges, and we've had to account for these and deal with many aspects that we didn't really anticipate. But just to kind of give you an overview, these obviously include all of the pre-sample variables like purification methods, where the samples come, the isolation tubes. We obviously want to isolate EVs from them, and something that's become very clear to us is we're interested in identifying EV subsets from this heterogeneous mixture of EVs to use them as prognostic biomarkers. So we've developed methods to isolate EV subsets, and then the next and biggest challenge really for us is the characterization. There've been a huge number of characterization methods, and they all have their pros and cons, and they all give different types of measurements. And something that's been incredibly hard for us to do is to bring together lots of these EV characterization methods and have a very concordant data set. So in some instances, this has been developed. We've built software that's allowed us to calibrate measurements so that we can perform cross-platform studies that have concordant data. And the idea is that we're trying to develop all of this so that we can start using the clinical trials here at NCI and see how much utility EVs have for their diagnostic and prognostic potential for cancer treatment. So the real hurdle in the field at the moment is concentration. Concentration is easily one of the most reported statistics in the EV literature, and it's likely also one of the least accurate, as most EV concentrations are reported as absolute concentrations without a limit of detection. And we can see over time, as our techniques have developed, that the reported concentration of EVs has increased. And this isn't surprising, as our assays have become refined, and we've also developed more sensitive technologies. But the important thing is that all of our instruments, even today, have limits of detection, and it's very important that we're able to define those limits of detection if we're planning to perform translational studies and validate one another's work. So inherently, when we report concentration, we're reporting the number of particles that we can actually detect. We can only report a concentration above our limit of detection. And diameter is, again, another very widely reported statistic. Often a mean diameter is reported, and comparisons between mean diameters is often reported in the literature. But the statistic is obviously skewed highly by the limit of detection. And you can see here with 3 limits of detection, we can get that only span 200 nanometers. Obviously the mean's going to span over 200 nanometers from the true mean. And this obviously has implications on concentration. So most translational studies, one of the outputs from them, and one of the ways that they usually quantify EVs is in changes of concentration of EVs. But the concentration that we detect and we report isn't necessarily able to be validated. If we report a bona fide change in concentration and we go to another instrument with a higher or lower limit of detection, we won't really be able to validate someone else's samples. And this again shows that from the true population of having this concentration, this is halved by having a limit of detection around 100 nanometers, and that's where a lot of detection methods at the moment have been to date. So this idea of absolute concentration that we've been reporting neglects all of the variables concerned with detectability, all of the light scatter measurements and fluorescence measurements for optical techniques, which have been highly utilized by the field, and something that we've been trying to promote. And really this is being promoted by a number of groups now within the extracellular vesicle community. It's becoming recognized that we need to define our limits of detection to standardize our literature. And ideally what we're working towards that's instrumental for our pipeline, but also for the field, is reporting concentration above a limit of detection. So the way we do this, and the way we have to do it for many optical techniques, is by doing calibration, and all instruments need to be calibrated, and performing calibration gives us the power to do a number of things, as well as defining the instrument sensitivity. We can define the instrument resolution if we have enough standards, and you can see here why that's important, because when we look at our cumulative signals like this, we wouldn't necessarily know we have 3 populations in a very low resolution instrument compared to a very high one, and most of our instruments actually vary in resolution. The resolution gets worse as they get closer to their limit of detection, and this is again why resolution is important to quantify, as it allows us to make traceable measurements. Another reason why calibration is critical, especially for techniques that have arbitrary unit outputs, is we can actually identify which instrument we want to use, which detection methods, which detector settings we want to use. And then we can also perform validations of our assays compared to other people's. So it's incredibly powerful to perform calibration, and it's something that we've been really pushing as a lab and many working groups in the field. And the reason it's critical, and this is an example of a proof of principle from flow cytometers of uncalibrated data here versus calibrated data, and with calibrated fluorescence in terms of molecules of equivalent soluble fluorophore, and the diameter has been calibrated using light scatter modeling. And these are two different instruments with two different limits of detection, and you can see on this LSR Fortessa, we're clipping part of the population. So if we were to report the concentration between the two, we would say that the concentration detected in the Cytoflex was higher. But if we wanted to make comparisons between the two instruments and we didn't have the calibrated units, we wouldn't be able to make a fair comparison. By calculating the limit of detection based off of our calibrated parameters, you can see that we're able to normalize these differences in detected concentration by the limits of detection. So this is really important for EV populations, especially because we don't have nice homogeneous populations that have tight CVs like this retrovirus. We're dealing with these kind of power law distributions, as Mike showed you in the previous presentation. So we don't really know any proportion of a population we're detecting. We're really reliant on our limit of detection. And we can only normalize that by having calibrated units. So the MyFlowCyte EV framework was developed for flow cytometers, and this took over 5 years to develop, and it was as a result of trying to perform standardization studies in the field and realizing that everyone has different instruments, and we couldn't get the same results. And the only way that we could get the same results is actually by accounting for all of these variables. And although this framework has been laid out for flow cytometry specifically, it isn't. Most of these actually can be utilized on other optical instrumentation. And ideally, this kind of framework would be developed for most characterization techniques, if not as a learning resource, but just to outline the things that we need to be aware of. And we can perform all of the controls needed to know that we're detecting single particles, but until we perform instrument calibration and define our instrument collection parameters, others won't be able to validate our data. So it's really important that we're transparent and we calibrate our data. So one of the case studies I wanted to show you is we have an internal reference standard in our lab group. It's these DC 2.4 EVs. And this is a cell line that we've grown and isolated EVs from because they're very efficient at producing EVs. And they were very useful for us to develop assays for. So we've developed these for sorting, for staining assays, and we've also used this same EV derivation to test every analysis technique that we've come across. And a few years ago, we were analyzing these EVs by flow cytometry. We could see a very nice tight population here that wasn't present with the noise. When we compared our flow cytometer concentration to our NTA concentration, they were very linear. We thought we were set. We thought we were detecting the majority of EVs, and it was very useful. And in the last few years, as detection techniques have developed, we've kind of learned a different story. So I wanted to just take you through this. So this is just how we've isolated our EVs. We deplete our FBS and grow the cells until they're 80% confluent. We concentrate our supernatant, our cell-free supernatant, and then we isolate EVs from protein using QEV10 columns. And in this case, we used the highest concentration of EVs, which was in fraction 7. So typically, NTA has been our workhorse for a number of years. And as was said at the start of this webinar, there's a reason it has been. It was one of the few techniques, 5 or 6 years ago that could robustly detect particles down at 100 nanometers. But as the field has developed and as more sensitive techniques have developed, there's been more time to scrutinize techniques. And at the moment, there's currently no demonstrated method of defining the limit of detection for nanoparticle tracking analysis. So this is a real problem for the reasons I outlined earlier. If we can't define a limit of detection, we don't know what proportion of the population we're detecting efficiently, and it makes it much harder to validate and standardize our results compared to different instruments with different limits of detection. So this was the kind of diameter distribution that we were used to with the NTA. And as we moved more recently to a flow cytometer with an APD, we saw we got this really nice tight CV population that we saw on our Astrios and our early assay development, but we also saw this tail started to develop. So this diameter, as I said, has been produced from using light scatter modeling, and flow cytometry light scatter modeling isn't a perfect technique for producing diameter distributions. You do have to make assumptions, but it's very clear that there was something else going on at this lower population. And then as we first tested the Spectradyne instrument, we were even more surprised to see that this population just kept on increasing. And this population that we'd been defining for a number of years and thought we were seeing the whole population with NTA and our lower sensitivity flow cytometer actually turned out to be the minority. And the nice thing that we have with flow cytometry and the resistive pulse sensing technology is we can draw a very clearly defined limit of detection. And you'll see why this is useful later. So after we'd performed both of these techniques, and we had an optical technique with flow cytometry producing this diameter distribution, we have a non-optical technique that's kind of validated this distribution. So regardless of assumptions, we're still getting the same distribution. And then we performed EM. So you can see that there are lots of these very small particles in here, and then there's a second population of these larger particles. So when we produced the diameter distribution, we actually found out it's bimodal. And there's another story that's actually behind this cell line specifically and the type of EVs it's producing. But the true EVs in the classical sense of just having a lipid membrane and a cargo that doesn't contain retrovirus or a capsid, which we think these larger ones do, appear to be much lower than 100 nanometers, down at the 60, 70 nanometer region. And EM obviously isn't something that we can do on every sample, and it's why it's important to have techniques that we can rely on for producing diameter distributions and make comparisons. So this is showing an overlay of all of these different techniques and normalizing them. And you can see that the Spectradyne and the flow cytometers both predict this second population, although they're not able to detect all of it. But it really highlights the importance of why we need to define a limit of detection. You can see here we are detecting particles lower than 50 nanometers with NTA, but there's no clear cutoff. So it's hard for us to normalize this data with respect to concentration to flow cytometry and resistive pulse sensing. And something that we're really trying to push and is becoming ever more evident is that the limit of detection really has to be defined. And a lot of the literature coming out now with high sensitivity techniques are showing that the vast majority of EVs are actually in fact below 100 nanometers, particularly in biofluids. And this is why it's important that we report a limit of detection so that we can make comparisons between each other's work. So with that, there's lots of people that contributed to all of our work, and I'm happy to take any questions.
Thank you very much to all the presenters. So, ladies and gentlemen, this is an excellent time to now pose your questions into the question box, and then the speakers will be happy to address them, the questions. So there are already a few questions. I'll lead off with one question for Jean-Luc, which is, there's an increasing interest in the use of microfluidics for studying circulating biomarkers. So in that vein, could you perhaps comment in light of work by Mehmet Toner and many others looking at circulating tumor cells and such, the applicability of your platform, the Spectradyne platform, to study different classes of circulating biomarkers?
Yeah, thanks. And thanks both to Josh and Dr. and Michael for excellent presentations. Thanks very much for contributing today. Yeah, so I'm certainly not an expert on CTC research, but the little I know is that they're very rare particles in complex samples, right? And so to that measurement challenge, Spectradyne has some advantages, which are that that we able to count many particles, so be able to in a reasonable amount of time. It's very fast measurement technology, and also it's capable of measuring very complex mixtures because the polydispersity of the sample does not affect the quality of the measurement. Getting at specificity of particles is a little more challenging with MRPS right now, I will say, but stay tuned.
Great, thank you. There is a question from the audience which says, why does the TS300 have a lower limit of 50 nanometers, and can it accurately measure anything lower? Jean-Luc, perhaps you want to take this?
Yep, sure. So we have different cartridge sizes that span different measurement size ranges, just because the way the technology is fundamentally any given cartridge design has a certain range of applicable sizes. So the TS300 cartridge measures down to 50 nanometers, and the TS400 cartridges span a different range with a different limit of detection. We are continually working to push the limit of detection smaller, as Dr. Paulaitis showed some preliminary results in his talk, that we have cartridges under development that can go down lower to in the 30 nanometer size range, but they're not quite ready for prime time yet.
Great. There's a follow-up question from the same as a part two. Why are cartridges not reusable? So perhaps Jean-Luc, you'd like to take that as well.
Yep, sure. The cartridges are designed for single use, and as part of the engineering in the cartridge that enables them to work in such complex samples. So the way the cartridge is designed, and there's a lot of information about this on our website too, and I encourage you to go and read more. But there's a filter embedded in the cartridge at the input, and so you can imagine we're flowing a complex sample all through a tiny bottleneck in the fluid flow. And implementations in the past, as far back as Beckman, as Wallace Coulter's, had just the whole sample flowing up to the constriction. And when you scale down to the nanoscale regime, that becomes a challenge because almost every sample contains larger particles, right? And those will block the constriction. So what Spectradyne has done is added a filter that's essentially just a network of small fluid channels, all made of the same material, in each cartridge, and that collects the large particles before they have a chance to block the constriction and allow much more complex samples to be run relatively smoothly. So that's one of the innovations of our technology. But that makes them, we can't guarantee reuse of them, although there are certain cases where that can be done.
Great,
Thanks.
Thank you. Yep, there's another comment followed by a question, and I'll read the comment out first. With the NCS1, the enumerated EV counts and concentration are highly dependent on how the background thresholding and noise floor is defined, which tends to get messier close to the LOD around 50 to 60 nanometers. That's the comment. And the question is, what do you recommend as best practices for defining this background noise? And I think, Jean-Luc, again, question for you, sir.
Sure, yeah, well, so first thing I would say is, it sounds like you're using the NCS1, and I would encourage you, anytime you have questions, to contact our awesome support team for help with method development or data analysis questions. We're always available. I think with any technique, there's some fine tuning required at the limit of detection, because that's right on the hairy edge, right? So we have techniques and methods available for helping you with that, and definitely please reach out to us for that.
Thank you. There's another question. It says, when calculating the EV concentration, should you include anything down to the lowest range of your instrument? So, Jean-Luc, again for you, I would believe.
Well, maybe this would be a good one for Dr. Welsh, actually, talking about the limit of detection and including that in the measurement report, or the reported concentration. Yeah. Josh, are you available to answer that one?
Yeah, sure. So the limit of detection. So when we're analyzing samples, if we have a sample set, we work out the highest limit of detection across the chip, and then so that the area that's the noisiest, to make a defined limit of detection for the whole sample set. And that means that if that was 65 nanometers across all of the chips, or 70 nanometers across all of the chips, we can have a consistent comparison of concentration. And we would say that the concentration detected was at X concentration above 70 nanometers. If you want to make that a traceable measurement, it gets a little bit more complicated, because you'll need to know how the resolution is changing at your limit of detection, how confident you are that 70 nanometers is 70 nanometers, or whether it's 70 nanometers plus or minus 5 nanometers. So with that, you need to run some more controls. But I think as a step forward for the field, just stating the detected concentration above a limit of detection is definitely a step forward, and has been shown for us to be very reproducible without the traceable measurements within the lab.
Great, thank you. There's another question, which is how to control for swarm detection if many small particles are around. Do you need dilutions? And perhaps Jean-Luc, you want to take this and address
Sure.
it?
Yep. So when you're operating our instrument, it's, the raw data is displayed on the screen and updated every few seconds. And it's pretty clear in the raw data whether the sample concentration is too high or not. And so you can inspect that, and if it's too high, it's clear because the particle events become overlapping, and there's a large fraction of events where it's overlapping. And it's just a simple matter of diluting after that.
Okay, thank you. There's another question. Do the cartridges use deterministic lateral displacement as the filter?
I believe that technology, I think that's referring to like an array of posts where different size particles go different directions than each other, right, than other different sizes. So assuming that's true, then the answer is no. What happens at the filter is it's essentially just a network of small fluid channels that are the same size as the nanoconstriction itself, as the sensing constriction. So it's much more likely that a large particle will stop at the input filter because it won't be able to go through the same constriction, and that reduces the blockage events of the constriction. Does that make sense?
Thank you. Ladies and gentlemen, if you have any follow-up questions, please feel free to reach out directly to Spectradyne, or you could send them to SelectBio, and we'll be happy to forward them over to Spectradyne. At this point, perhaps I will ask Jean-Luc to make some closing comments regarding the technology and how to get in touch with them prior to us closing the webinar.
Yep, great. Thank you very much, Yanal, for organizing a good webinar, and especially to Dr. Welsh and Paulaitis for joining us and sharing some of your exciting work with us. I think that we need to be listening to these people because they're real experts in the field and have good broad understanding of where the field is for EV quantification. So just thank you very much. And if you want to learn more, we welcome you to email us or visit our website for more information. Thank you very much.
This concludes the webinar. Thank you to our speakers. Thank you to the sponsor, Spectradyne, for excellent webinar. And ladies and gentlemen, please stay tuned for more webinars coming your way from SelectBio. Thank you, and have a great rest of the day. Bye-bye.